So far, we have focused our lecture on information that can be learned from protein structures for individual proteins. In this section, we will quickly review which properties can be calculated at the amino acid residue level, at the protein scale, and what the changes in these properties can tell us at the genome scale. This section will put the systems in structural systems biology. As we learned in an earlier section of this lecture, each amino acid residue has different R groups, which lends to distinct physiochemical properties between the 20 amino acids. As we zoom out to the secondary structure, we can identify which amino acids in a structure contribute to helices, turns, sheets, as well as additional rarer secondary structures. Zooming out to the tertiary structure, the final shape of independently stable domains, we can look at the solvent surface accessibility of any protein. Simply, are the residues present on the outside or buried in the inside of the protein? Furthermore, we can analyze the amount of bonding within the protein interior that can help determine its stability in different environments, such as pH stress or temperature stress. Focusing on the active sites of proteins, substrate specificity and kinetic parameters can be estimated using methods such as molecular docking or measured in vitro. Some properties of proteins can be predicted in silico. We already discussed using homology methods for predicting structures when we cannot find an experimental one in the PDB. Additionally, two or more structures can be aligned in 3D space. This is called structural alignment. It can be used to determine how similar folds and domains, which you should remember are conserved patterns of protein shape, are. These calculations can help us predict the function of domains that have not been experimentally verified. Additionally, we can predict interactions between proteins and small molecules or even other proteins. These docking techniques have many applications in drug design. Lastly, we can also predict the movement and activity of proteins using molecular dynamics and other simulation methods. The important thing to remember is that each of these prediction methods has a different accuracy and costs associated with it, with some of these methods being very computationally intensive. The slide deck will have more slides regarding predicting properties in silico, but the video will not focus on these. Feel free to look through the slide deck now for additional resources. Calculating some or all of the protein properties we talked about for an entire genome can give us a genome-scale model with protein structures, or GEMPRO for short. From a systems biology perspective, we look at cells as a network of reactions the cell genome is capable of carrying out. For each reaction, there is an associated gene and logically a protein that carries out this reaction. Mapping all the proteins in the network to a protein structure is how we create GEMPROs for different organisms. Each protein property is mapped at the amino acid level. Then, a GEMPRO maps these properties, along with properties unique to the final shape of the protein for all proteins in the network. Think of it as a matrix within a matrix for proteins mapped at the residue level, while just the matrix on the bottom right for proteins and their global properties. The basic workflow of any GEMPRO is to identify all the genes in the organism, map their amino acid product to the PDB database, or use homology methods such as ITASR to determine what the protein structure is. Since sequences can map to multiple structures, the structure is refined and the best one is chosen. Using these GEMPROs, we can now compare protein properties within an organism's own reaction network or between multiple organisms. Let's focus our attention on some applications of GEMPROs in the past. The slide deck will have many more applications of GEMPROs than will be covered in this video. Please refer your interest to these additional slides. The structural genomic efforts of the 1990s yielded tons of new protein structures, namely for the organism Thermatoga maritima, an organism that lives at very high temperatures. Mapping these structures onto the metabolic network of Thermatoga resulted in the first ever GEMPRO. Now, within a metabolic network, there are reactions scattered throughout that are essentially the same, even though they are far apart from each other in the network. These are called similar reactions. Here, we see that ARGB and lyse both phosphorylate their substrate using ATP. The next reaction, coded by the genes ARGC and ASD, are also similar. Reactions that occur sequentially, such as ARGB-ARGC reactions, are called connected reactions. In the evolution of metabolic reaction networks, in this case Thermatoga, it was unknown if the protein responsible for one reaction evolved such that it could now catalyze the next connected reaction within its own pathway. This would extend the reaction network one reaction in the series. The other hypothesis was that proteins conducting the similar reaction would be mutated and over time recruited to fulfill the next sequential reaction need. By comparing the shape or fold similarity between proteins within connected reactions or similar reactions, the latter hypothesis held true. This means that protein and connected reactions are much more different from each other, even if they are right next to each other in the metabolic network, than they are from proteins in similar reactions that are more distant across the metabolic network. The first example compared proteins within an organism's own metabolic network. Here, the same GemPro of Thermatoga from the previous example was compared to one created for E. coli. By comparing the protein properties between the two organisms, Elizabeth Brunk and others identified that the proteome of Thermatoga has many more buried nonpolar residues than does the same protein in E. coli. Why would this be? Well, if you are paying attention earlier in this section, buried nonpolar residues can help stabilize the protein's interior. Since Thermatoga lives at much higher temperatures than E. coli, it is reasonable to assume that the proteome overall of Thermatoga uses this increase in buried nonpolar residues to maintain thermostability of its proteome at high temperatures. The first two examples focused on protein properties, but another aspect of structural systems biology is mapping functional information onto these structures. Here, a human GemPro was created. Information regarding point mutations associated with various cancers were overlaid onto this GemPro. That is, that each mutation was mapped directly onto its corresponding amino acid in each protein structure. If you remember, we discussed that shapes of proteins are found in patterns that can be relatively conserved. Here, the structures that shared conserved domains were aligned. This allowed for all the information contained within the domain of one protein to be directly compared in 3D space to the information contained in another protein with the same conserved domain shape. Thus, mutations in one gene could be directly compared to mutations in another gene. Using this method, 3D hotspots of mutations associated with disease were elucidated, even though these mutations had occurred in separate genes. Rather than associating the disease state with a particular gene mutation, protein domain mutation associations can be made and might help elucidate a mechanistic understanding of disease states. Questions such as, how does a specific domain contribute to the disease state once it's been mutated, are on the horizon. Let's look at how protein structures can be used to guide constraint development in systems modeling. Briefly, using the protein structure, the thermostability of each protein was determined. Then, a temperature-dependent model of kinetic efficiency was calculated for each protein. In a metabolic model, flux is optimized through a reaction network to solve for a growth rate. These fluxes through each reaction are constrained by the bounds on the kinetic parameters of the reaction. Thermostability was used to change the kinetic bounds for each reaction in a temperature-dependent fashion, thus constraining the model. This allowed for the prediction of temperature-dependent growth rates that matched experimental data, further increasing the level of detail with which M models can predict biological processes. In a ME model, a genome-scale model of metabolism and macromolecular expression, the E in ME stands for expression. Like in an M model, the presence or absence of a gene determines whether or not a reaction associated with that gene is present in the network. Unlike M models, however, ME models contain reactions that include transcription and translation. Therefore, for each metabolic reaction to be present and active, the gene or genes responsible for it must first undergo their transcription and translation reactions. If a protein is very large, it will require more substrate allocated to its synthesis. This gives us a synthesis cost for each protein. These models predict protein expression as well as growth rate. Using a similar approach to the previous example, the thermostability of the proteins were calculated. An additional chaperone network was added to the model. Chaperones are a group of proteins that are responsible for maintaining the proper folding of other proteins in their functional state. If a metabolic protein is more unstable at any given temperature, more chaperones are required to maintain it. Thus, this model could predict not only the temperature-dependent growth rates, but also the temperature-dependent proteome allocation, namely, the amount of chaperones the cell needs at any given temperature to maintain maximal cell growth rate. Here is a brief list of papers that use GemPros. Feel free to pause the video and explore these papers in detail. Finally, let's take a look at some avenues for the future of structural systems biology. We will skip the obvious ones, such as developing GemPros for more and more organisms as their structures become experimentally determined. The first roadblock to widespread usage of GemPros is that they are very difficult to recreate and use. There are many steps to map sequence to structure and many dependencies on softwares to identify protein properties. Our lab software, SSBio, which stands for Structural Systems Biology, is the first step in this direction. However, its integration with other data types requires additional organization to maximize its usefulness. Here, by creating a database that integrates data from multiple disciplines in an easily accessible format, such as a CSV file, structural properties can be more readily used by both systems modelers and biologists, in addition to structural biologists. The goal is simple: organize a database that can track a specific amino acid all the way to the system scale. In the blue box, every reaction in the network for E. coli has been mapped to its multi-subunit structure. This section is organized in a way that preserves the gene-enzyme stoichiometry associated with each reaction. In the second box, the purple box, all the structural properties for a given structure have been calculated. This includes basic amino acid properties from the primary sequence all the way to the quaternary structure, mapping genes to their quaternary protein structure rather than their tertiary structure as did previous GemPros. Properties such as membrane integration and protein-protein interfaces can be mapped. Lastly, all functional annotations were mapped to each protein. This includes information such as where active sites are located, which regions bind DNA, RNA, metals, and others. Additionally, information regarding the phenotype of studied point mutations was mapped, as well as mutations from our group's adapted laboratory evolutions. By integrating all these data types into one easily accessible data frame, the use of structures for developing model constraints as well as for understanding or interpreting mutations on a genome scale will be much easier. Since the database described on the previous slide studies protein structures at their quaternary level, the transmembrane residues can be mapped onto the structures and used to calculate the surface area required for each protein in the membrane. Additionally, the volume of each protein can be calculated. This allows for the compartmentalization of the cell proteome of E. coli at the angstrom level. As you may recall, ME models predict the proteome expression or allocation at the genome scale. Using spatial constraints, namely size and volume, the physical space taken up by each protein in the cell can be calculated. On the right, we show proteome maps comparing the protein expression levels with the physical space they take up in each compartment. This can have a wide variety of impacts on cellular phenotype that has yet to be explored. Another future avenue for structural systems biology is to work with visualization tools to model and visualize the entire cellular proteome. Methods, such as cellPACK, can be used to pack these optimal cell proteomes. Other questions remain. Can we model the movement of proteins within a cell using molecular dynamics simulations? Can we compartmentalize the cell packing and subsequent simulations? Or can we use these image tools as educational resources to disseminate the findings of our genome-scale models? New avenues for structures in functional biology build off the ideas of mutational hotspots. In addition to identifying hotspot regions of mutations, as in the case of Recon 3D, structural statistical methods for measuring the significance of these hotspots can offer insights into regions of protein function which were previously unknown. For example, if we identify many mutations in a region that are statistically distinct from random, it may signify the functional importance of previously unknown domains. Additionally, if multiple mutations are distributed throughout a structure, can we group these mutations in statistically significant clusters? And can we study the mutations within a cluster? Do they have the same phenotypic effect in each cluster? Are the phenotypic effects different between the clusters? Questions such as these and their answers may soon enable rational design of protein structures at the genome scale. Lastly, structural systems biology has many promising implications in metabolic engineering and synthetic biology. Scaling up the angstrom level knowledge we gain from the protein structure, it may soon be possible to conduct enzyme engineering across an entire genome. By combining systems modeling with structural biology, we may soon be able to identify bottlenecks in the metabolic network, as well as rationally design better enzymes to overcome these bottlenecks for every node in a reaction network. Thereby, structural systems biology may soon be improving bioproduction yields for existing byproducts or even rationally designing pathways for producing new bioproducts. In this lecture, I hope that you have begun to appreciate the wealth of knowledge and the level of detail protein structures offer. We have also shown that technological advancements in determining protein structures have made it feasible to rather quickly obtain high-quality images of larger proteins. Surely, the first complete structural proteomes will soon be experimentally available. We have also seen how structural systems biology can be used to answer biological questions and gain knowledge at the genome scale. And lastly, we talked about the many unexplored opportunities that still remain. That concludes this lecture mini-series. Thank you for your time, and remember, wash your hands.